The cell cycle is the life cycle of a cell from the time it first forms until the time it divides. One cell divides into two through a process known as cell division. Parent cells divide. Two daughter cells are genetically identical or nearly so to the parent cell. It enables growth and the replacement of damaged cells. Prokaryotes produce through binary fusion, whereas eukaryotic cells divide through either mitosis or meiosis. Interphase is the first part of the cell cycle. It's the phase in which the cell grows and the DNA replicates in preparation for mitosis. Interphase occurs in three subphases. There's the G1 phase, which is the first gap in which the cell grows, the S phase, which is synthesis, in which the chromosomes are duplicated, and there's the G2 phase, which is the second gap in which the cell grows more and prepares to begin in the process of mitosis.
In interphase, there is the nucleus filled with duplicated chromatin, the nucleolus, the nuclear envelope, and centrosomes. After interphase, the mitotic phase begins.
The mitotic phase is a 5-stage process in cell division that produces exact copies of the parent cell's chromosomes and segregates them into separate nuclei, followed by cytokinesis to produce two daughter cells.
During prophase, the chromatin condenses into chromosomes. The mitotic spindle begins to take shape. The nuclei and ribosomes disappear.
During prometaphase, chromosomes condense further. The sister chromatids bind kinetochores. Kinetochores attach to spindle fibers.
In metaphase, chromatids line up along the cell equator.
During anaphase, sister chromatids pull to the opposite poles. Proteins attached to the chromatids are released, causing the sister chromatids to rapidly separate.
The final stage in mitosis is telophase. The nuclear membrane reforms and the chromosome segregation is complete. Shortly after mitosis, the cytoplasm finishes dividing throughout the process of cytokinesis. The system that controls which phase a cell is in is called the cell cycle control system. The times at which the cycle can be stopped or pushed forward are called checkpoints. In these checkpoints, the cell decides whether it is safe to proceed or stop and not divide. Checkpoints include the G1, the G2, and the metaphase checkpoints. The most important checkpoint for mammals is at the G1 phase. If the cell isn't given the go-ahead at the G1 checkpoint, it goes into G0 phase and stops dividing.
When things go wrong at the checkpoints, the cells keep dividing. This can result in a tumor or cancer. Breast cancer occurs when there is uncontrolled growth of abnormal cells in the breast. Old or damaged cells won't stop dividing, causing a malignant tumor. Mitotic chromosomal instability causes tumor progression. This is due to abnormal mitosis. When things go wrong at the checkpoints, abnormal mitosis can occur. According to a study done by Meng Erhang and colleagues, patients with platinum-resistant ovarian cancer can be resistant to chemotherapy. This is due to altered regulation of the cell cycle checkpoints.